Okay, so we're going to be doing some enzyme digestion experiments, 4 of them in total. The first one is going to be involving fat digestion. The fat we're going to be using is called litmus cream. The enzyme is called a lipase. So most enzymes end in ASE, as you'll see. This enzyme's job is to basically work on litmus cream, and whatever an enzyme works on is called the substrate. So our substrate today is called litmus cream, and it's sort of purplish in nature to begin with, which means it has a pH indicator. So if this fat breaks down, and the enzyme helps to do that, then the resulting product, which are fatty acids, should turn this purplish cream somewhat red. That's what we're expecting. So we're going to set up 4 different tubes, all of them containing litmus cream, each one containing a different aspect of the enzyme or its helpers. The tube number 1 basically contains just the litmus cream, no enzyme, no helpers. Tube number 2 contains the litmus cream and a compound called bile, which you find in your digestive system. What it does is it helps the enzyme, in this case called a lipase, to break down the fat. The third tube is going to contain the litmus cream and just the lipase, but no bile. So it has no helper here in the third tube. The fourth tube is going to contain everything, the litmus cream, the lipase, as well as the bile. So the enzyme, in order to work well with its helpers, usually needs to be incubated at a somewhat higher temperature, like 37 degrees, which is about body temperature. Below that or above that, it doesn't do too well. So what we're going to do is we're going to take these 4 tubes and incubate them at 37 degrees for an hour. Okay, so it's been an hour. That's sufficient time now for the enzyme to have done its job. In this case, the enzyme is the lipase, and the substrate, what the enzyme works on, is called the litmus cream, the fat. And so it's been an hour at the proper temperature, 37 degrees. So let's look at the 4 tubes. Tube number 1, which had no lipase, no bile, just the litmus cream, we should expect nothing, and it still remained purple. Tube number 2 has the litmus cream and just the bile, but no enzyme. So we see a tiny bit of change, but not too much. Still purplish. Tube number 3, we had the litmus cream and just the lipase, no helper bile. And so we see somewhat of a change in color, but not much. Tube number 4 is where we have everything, both the lipase, the bile, the litmus cream, and we should see the most change here, and we do. We see a deep red color, and that means this fat, the litmus cream, has been broken down into fatty acids, which is exactly what we expected. And that has changed the purplish color to red.